As the title says, I'm going to talk about two pore domain potassium channels and their potential as therapeutic targets. So you've already heard from Stephen yesterday, Stephen Tucker. Oops. Let's find the right button. The family of mammalian two pore domain potassium channels, there's really 15 different members that can be divided broadly into 6 subfamilies based on both their structural similarities and certain functional similarities. All of them have characteristic of 4 transmembrane domains and 2 pore regions, 2 P regions, and so it's envisaged that they come together as dimers, contributing 4 P regions to the pore. And there's increasing evidence that more and more of them, at least within the families, can form heterodimers, which adds to the complexity. That's certainly true for the TASC family. It's looking as though it's also true for the thick family and the TREC family as well. So that's quite a complication. So over the past, relatively recently, over the past 2 or 3 years, there's been a number of different studies proposing for quite a number of these different channels roles in pathophysiology and potential disease states. For example, TREC1's been implicated for quite some time actually in depression, TRESC and migraine. Interesting paper a year or so ago on TASC3 and cancer, TASC2, KCNK5 and deafness. And then the 2 I'm going to focus on actually down at the bottom here are a syndrome called KCNK9 imprinting syndrome, arising because of mutations in TASC3, and then pulmonary hypertension, where at least some cases are due to mutations in TASC1. So KCNK9 imprinting syndrome is known, or at least in the original cases identified, is due to a single point mutation in the pore region of TASC3, KCNK9 TASC3. And this occurs at an amino acid which is 5 amino acids towards the C-terminus of the channel from this conserved hinge glycine residue that's present in virtually all potassium channels. Most potassium channels have a small amino acid at this residue, including TASC3, but 1 or 2 of the K2P channels and 1 or 2 other channels don't. They have a more bulky amino acid here, and that does interfere with certain regulation of the channels, certain gating properties of the channels. But for TASC3, normally this is a small amino acid, a glycine residue, but in the disease state, KCNK9 imprinting syndrome, it's called imprinting syndrome because it's an imprinted gene, and so we inherit TASC3 from our mothers, or mostly from our mothers. So it's much more susceptible in that sense to point mutations. And mutations here at this amino acid, changing it to an arginine residue, is what's been identified in patients that suffer from KCNK9 imprinting syndrome. So KCNK9 imprinting syndrome, what is it? Well, from the original description, 2008, all affected individuals identified in this paper had moderate to severe mental retardation and were hyperactive, severe feeding difficulties at infancy, dysphagia of solid foods until near puberty, and then generalized hypotonia at an early age, followed by weakness of proximal muscles. And then the original description of this syndrome, the authors, Israeli group, did what you would like to do, and that is created the TASC3 channel with this mutation in it. And as far as they could see, doing oocyte recordings, there was no functional current through these channels. So they described it as a complete loss of function mutation. And one of the things that's known to happen as a result of this mutation at the very early stages of development is there's a dysfunction of neuronal migration. And this is just an example from the cortex, paper from a couple of years ago now, where we're looking at migration of neurons, cortical neurons, to layer 2 and 3. And this is impeded either by a mutation in task3 at the pore region, which stops the channel being functional, but also by this point mutation here found in this disease, KCNK9 imprinting syndrome. So a few years ago, we revisited this mutation for a variety of other reasons. And we actually got quite surprising results. So these are, I'll just take you through our current traces here. These are currents through wild-type task3 channels using the kind of unusual protocol that we use to try and get maximum information from a single sweep, both a step and a ramp in the change in the voltage. Current voltage relationship here, typical for a K2P channel. But when we looked at the KCNK9 imprinting syndrome mutation, this G236R, although the current was substantially reduced compared to wild type, it wasn't abolished. There was still a functional current there, but it had slightly unusual properties. Now it changed from being effectively an outwardly rectifying current to an inwardly rectifying current. And you can enhance the current through this mutated channel by doing experiments, for example, in symmetrical potassium to increase the driving force for the current. We see quite a large current now through the mutated channel, but it's still inwardly rectifying, even in symmetrical potassium solutions. And that's probably, the inward rectification at least, is probably because we're introducing a charge residue to the inner pore helix here, interfering with the flow of ions through the pore. However, we have found ways of reversing, in inverted commas, this mutation. So by coincidence, the amino acid immediately next to this one has been known for a while to be a gain of function mutation of task3. And by doing the double mutation, we can actually restore current to virtually wild-type levels through these channels. So we can get a gain of function mutation to reverse most of the effects of G236R, although it still retains its inward rectification. It's quite difficult to get compounds that activate task3 channels. We're still wrestling with this in collaboration with others. But one compound that we've used occasionally that's not a great enhancer of task3, but it's one of the only ones we've got, is flufenamic acid, afenimate. And it causes a small enhancement of current through wild-type task3 channels. But interestingly, for the mutated channels, we actually get a much bigger effect of the compound. It's a much better enhancer of current through these channels than it is through wild type. So we can also, as well as getting genetic recovery from the mutation, we can also see pharmacological recovery to a certain extent of the mutation. So the G236R mutated channels give rise to this syndrome. Mutated channels carry significantly less current than through normal wild-type channels, and they show inward rectification. However, current can be restored, at least in part, either by a gain-of-function mutation or, more interestingly perhaps, pharmacologically. Now, I didn't for 1 minute think this would be something that we could suggest as a treatment or as a potential treatment for the disease, but more and more people are being identified as having this syndrome, and this is Preston Kelly, who lives in California, and his mother, Melissa. And Melissa, ever since Preston was diagnosed a couple of years ago, has been a very strong advocate for further research in this area and doing everything she can to help her son and to help everyone else suffering from this disease to get the best treatment that they can. And although flufenamic acid isn't available for use freely in the States, a related compound, mefenamic acid, is. It's called Ponstel in the USA. And Preston's clinician has been treating Preston for over a year now with MFA. It does also enhance current through this mutated channel, although not nearly so well as FFA. And they feel, although it's a very small sample number obviously, that his development responsiveness is clearly better while he's on MFA compared to when he's not on the drug. So now we have a database, really working with our colleagues in California, of about 30 individuals that have clinical syndromes consistent with KCNK9 imprinting syndrome and have undergone genetic screening, so they all have a mutation in TAS3, an identified mutation. Most of them, 80% of them, have the same mutation, G236R, but about 20% of them, 6 of them, have a different mutation in the channel. And interestingly, which makes life slightly more difficult for us, for at least some of these mutations, current isn't reduced. and doesn't show inward rectification, but the regulation of the current by other things is altered. So it may be that for some of patients, the right strategy is not to directly enhance current, it's to look at the way it's regulated. So I'll skip on to the second thing I wanted to talk about, and this is changing to a different K2P channel related family member, task 1, KCNK3, and its suggestion as a potential therapeutic target in diseased pulmonary hypertension. Pulmonary hypertension is also a rare disease, not so rare as KCNK9 imprinting syndrome. I think on the average globally, about 1 in 20,000 people suffer from pulmonary hypertension, but its incidence is vastly increased if people are suffering from other diseases, other infections. And for example, and quite interestingly related to Jamal's talk just before, people suffering from HIV infection have an increased risk of pulmonary hypertension up to 1 in 200, so a 100-fold increased risk of developing pulmonary hypertension following HIV infection. And our interest in this was kind of piqued by a paper from 3 or 4 years ago now in the New England Journal of Medicine showing a number of different patients that had mutations in task 1, KCNK3, and had developed pulmonary hypertension. And this group went on to show that, similar to the experiments I showed you with task 3, that they could, first of all, with wild type task 1, that's the gray trace at the top here, they could get enhancement of current with a compound called ONO-RS 082. So this is an activator of task 1 channels. Has a slightly unusual, I'll come back to this slide, but it has it's used at the moment as a phospholipase A2 inhibitor. This is its structure, but we believe, although the evidence is not completely compelling, that it is actually acting directly on the task 1 channels to cause enhancement. And interestingly, this group found that for at least some of the mutations that were underlying pulmonary hypertension, they could get recovery of current through these channels by applying this compound. So they could get pharmacological reversal of a mutated channel. We collaborate with a group in Madrid, and they just, at the end of last year, published a paper where they'd identified 2 new mutations in task 1 that lead to a very severe form of pulmonary hypertension in children in Spain. These are the mutations here. They're just at the top of the 2 transmembrane domains, transmembrane domain 2 and transmembrane domain 4. So in collaboration with our colleagues in Madrid, we created these mutations, and top just shows our data with wild type task 1. We can get the same effect of enhancement of current through wild type channels with this compound. But unfortunately for both mutations that we've looked at in the Spanish families, both of them show no current, and it can't be reversed pharmacologically. So in these particular mutations, we cannot reverse the effect by this particular pharmacological treatment. So it's going to vary depending on the mutation. Perhaps more interestingly, though, in the more general area of pulmonary hypertension, this is some data from a French group published towards the end of last year, Marc Humbert's group. And what they're looking at now is idiopathic pulmonary hypertension, so not related to a particular genetic mutation. And what they find in patients, in humans, suffering from pulmonary hypertension that hasn't got a known genetic origin, is that they see quite a substantial reduction in task 1 expression, although not a complete abolition, reduction in protein level, and also a substantial reduction in current. So this suggests that perhaps a really viable therapeutic strategy is to target task 1, not just in genetically mutated, in patients suffering from a genetic mutation, but also in the more general idiopathic form of the disease. So for the last couple of minutes, I just want to, have I gone too fast? Derek, I need to go back and do some more. I just wanted to pose some questions because it's a bit of homework, if you like. in the, both of the examples I've given you is where we're looking for activators of K2P channels. And the more I think about activators, the more grief it's causing me, because I've realized that I'm not actually clear how to quantify the effects of them so we can do viable comparisons with other studies. So I'll just talk through a couple of examples. So how do we best quantify activators of K2P channels? We're not just looking, for example, for 100% inhibition. Could get 110% enhancement. So do we measure the EC50? Do we measure the fold increase in current? Or do we measure it, and this is probably my preferred way forward, is to measure everything relative to a standard compound. And this kind of came to a head for me a couple of years ago when I was putting together a review of activators of TREC1 channels, so a different family of K2P channels, which are potentially useful in pain and a variety of other clinical disorders. On reviewing the field, I found that it was very difficult to actually compare one study with another. Some people described the fold increase that they would get. Some people described an EC50, like us. Some people did both. But there wasn't much consistency across the field, and it was hard to get comparisons between studies. And I think this, particularly for a leak current or a constitutively active current, this becomes an additional problem, if you like, is how do we deal with varying baseline constitutively active current across platforms? So you might have a different level of constitutive activity, baseline activity, in a flux assay versus a patch clamp assay, or in a mammalian cell versus an oocyte. And how do we deal with different activator effects between different channel isoforms? So splice variants of the same channel, or in the examples I've shown you, channelopathies due to mutations in the coding region. And I'll just give you one example of the problem as I see it. And this is TREC, an enhancer that Tony Lewis talked about yesterday in terms of fungal channels, BL1249, which is a good enhancer of TREC currents. This is its effect on wild-type TREC1 current at about a 50% effective concentration. But TREC1 doesn't just exist in one form. It has an alternative, a process called alternative translation initiation gives rise to a shorter form of TREC1, starting with this methionine here. So there's a short form and a long form of TREC1 expressed in different places throughout the body. The short form of TREC1 is quite different in its properties, in some of its properties, to the long form. So it's a much smaller current compared to what you might call wild type or the long form of TREC1. And it's also not completely selective for potassium. It has a reversal potential of around minus 47 millivolts compared to minus 85, suggesting some significant sodium permeability. But if we then look at an activator, BL1249, again on this short form of TREC1, what we see is that it causes, on paper, a massive enhancement of the current. So if you look at the current trace here, we're seeing a really substantive enhancement of the current with this compound. And in terms of a percentage enhancement, it looks as though this compound is acts much better on the short form of TREC1 than on the longer form, because the percentage enhancement is about 1,000 compared to about 200. But most of that is just down to the fact that the baseline level of current is much smaller to begin with. And so what's the right way to deal with this? Are we really saying that this is a better compound at these channels? Or is it, do we need to find a way of normalizing the effects of all compounds on each of the different isoforms? So I don't have a clear answer. I'm wrestling with this, and I don't know the best way to do this, but I'd really benefit from any input or advice either now or in an email at some point in the future, because we really need to sort this out if we're going to start screening for new activators of these channels. We need to have a standardized way of doing it. At the very least, a compound that most people agree is a reference compound that you can then compare effects with. So I'll stop there. And just thank my colleagues both at Kent and elsewhere for the work I've shown today. All of the work on task 3 was done by Emma Veal, and all of the work on task 1 was done by Kevin Cunningham in my lab, and we benefit hugely from our collaborations in the States and in Spain. Thank you very much.
Do we have time for some questions for Alistair? In the case of the imprinting syndrome, is there potential therapies that reactivate the paternal gene? Would that be a way of looking at it as well? And secondly, is task 3 also imprinted or not?
Is
task 1 imprinted as well?
No, task 1's not imprinted. It's quite rare for. I don't think there are many ion channels that are imprinted. There's 1 or 2, not many. Another confession of lack of knowledge is that I'd understood imprinting to be quite black and white, quite all or nothing, that you only inherited from one of the, either maternally or paternally. But recently I've been reading some stuff, including some work on task 3, where, in fact, if you go down to the level of individual neurons, you can find examples of
mosaicism.
Yeah, exactly.
So then, in the case of task 3, I guess that while you're looking at full mutants, is actually what's being expressed in cell is a mixture of pairs of wild type, non-mutated, and then only 25% of the cells will both have subunits that contain the mutation. Others will have one mutated subunit, wild type and wild type. So I guess it's a proportion of these that's expressed.
Yeah, I think in quite a large number of neurons, and I can't give you the detail because it's not our work and it hasn't been published yet, that you do only see what appears to be task 3 coming from the mother. And so if that was mutated, all of it would be mutated. Right. But in some neurons, some other neuronal types, much fewer neuronal types, you get more of a mixture, more of a mosaic. But we have got some work on forced concatenamers between a mutated form and a non-mutated form, and it still shows inward rectification in many of the properties that we see here, although not quite so severe. Yeah.
Hi, Alistair. So I just wondered whether for task 1, any of the mutations affected trafficking. Is it purely gating, or is there some trafficking component as well?
For the ones that we've got, the 2 that we've got, it's all about gating as far as we can see. We can't see any differences in trafficking. I think that's true also for the ones in the earlier paper, but I'm not. We haven't repeated that.
It's a great talk, and thank you for mentioning cancer in relation to.
Oh yeah, I did.
As we warm up towards.
It was an oversight. No.
As we warm up towards the ion channels and cancer session next, I've just done a PubMed search on imprinting syndrome and cancer, and over 100 papers come out.
Ah, okay.
So, are you aware of any possible connection through the channel?
Well, there's quite good study a year or so ago of the importance of task 3 in certain forms of cancer, but it's not something we've done anything on at the moment. But I think that would be really an interesting area to develop in the future, certainly.
Alistair, is it only the phenamate class of non-steroidal anti-inflammatory drugs that have this activator effect, or do other classes emulate?
No, the only ones that we've found so far are the phenamate class. Yeah.
Hi, I was wondering if the activity of the phenamates is that causing a change in the pore or single channel conductance, and if it's pore, could pore be the thing to look at instead of current augmentation? Yeah,
so Stephen Tucker could probably answer your question at the single channel level better than I can. But I think I think so. I played quite a negative hand there in terms of all the problems with how do you quantify these effects. But when I was thinking about it in relation to the way people quantify G protein coupled receptors, which is similar sorts of problems, and they use black box approaches. The one advantage we've got in the single channel field is that you can actually, in theory at least, look at the behavior of an individual channel and measure an absolute open probability. And so I think, although that wouldn't be trivial, to match something like that, so you can get a feel for where is the open probability of the channel, what is the maximum you can get with BL1249, has it still got 80% to go, for example, or can you never get it higher than that? But at least that gives you some ground rules that you can base all your other numbers around in a way that, at the moment, this G protein coupled receptor field can't do.
Yeah, I was thinking about Steve's neuron paper, the truncation. I don't know if you know what the PO was for the truncation, because if it was a lot lower, then
I think it was lower. I think in our hands we find it much lower than they found it. But
so probably further to go then.
Yeah.
So maybe I can ask the last question, which is more kind of clinical thing. So the way you presented it in terms of the clinical profile, it sounds like it's quite broad from mental retardation, hyperactivity. Is it known where these channels are expressed in the CNS? And,
well, they're highly expressed.
What role they have in circuit behavior?
Yeah, they're clearly expressed in the data I showed from the cortex. They were originally described as highly expressed in cerebellum, particularly. Although there is some difference between in the granule cells and the cerebellum. There's higher in rodents compared to humans, I think. But I think there's quite widespread expression of TAS3 in the CNS, much more so than in the periphery.
In the hypotonia?
Yeah, I'm not completely sure about that.